Hi, everybody. Thank you for taking the time out of your day to attend our presentations. My name is Joelee Tooley, and I am an MPS candidate participating in the Scientists in Parks Fellows Program for the 2021 season. Today, I will be talking about my project titled Climate Change Impacts on a Zoonotic Disease in an Alpine Ecosystem. Just a quick order of events for this presentation. First, I'm going to tell you a little bit more about myself. Then I'm going to tell you more about the park that I was working in this summer, which is Lassen Volcanic National Park and is pictured here. Then I will discuss the focal species of the project, the American pika, and to give you some details about the goals of my project and go through the actual field protocols and processing the animals that we catch. And then I will go over a brief summary of my data analysis and give you some of my preliminary results. And lastly, I'm going to discuss the impacts of the Dixie Fire on my project and the park, and give a quick thanks and take some questions. So a little bit more about me. I attended the SUNY College of Environmental Science and Forestry, which is a small research institution in New York State and is the oldest, most distinguished institution in the United States that focuses on the study of the environment, according to their Google description. I graduated from ESF in December of 2020 with a bachelor's degree in wildlife science, and I obtained my associate wildlife biologist certification through the Wildlife Society as well. I loved ESF so much, I decided to go back and do grad school there. I am currently enrolled in the MPS program, which stands for Masters of Professional Studies in Fish and Wildlife Biology and Management. So this is a non-thesis track master's, which basically just means I am more focused on applied science and wildlife management instead of primarily research. So my main area of study is mammals, and I am specifically interested in mesocarnivores, so things such as weasels, foxes, bobcats, even bears, stuff like that. And I'm also interested in wildlife disease ecology, which really matched up well with this project. In my free time, I love hiking and backpacking, seeing new places, hammocking around, cooking, and spending time with my two adorable cats. So my project took place in Lassen Volcanic National Park, like I said, and this park covers about 106,000 acres. So it's one of the medium-sized parks. And the park is located in Northern California within the Sierra Nevada mountain range. This park was established in 1916, which makes it the 9th oldest park in the United States. And most of the park is roughly at 8,000 feet in elevation, with Lassen Peak going up to 10,457 feet. The park is most well known for its bubbling mud pots, pools, steam vents, and fumaroles, which are all a product of the volcanic and geothermal activity that occurs in the park. And personally, I think it is a gorgeous park. It's truly a hidden gem, and you should go visit if you get the chance. And this red circle here at the bottom left corner is where my project took place, in the southwest corner of the park, also known as the Bumpus Hell area, where the majority of the geothermal activity is. As you can see in the picture on the top right corner, that is Bumpus Hell. So moving on to the species of interest, the American pika, also known as Ochotono princeps. The pika is a small mammal in the same order as rabbits, which is where it gets its nickname, the rock rabbit. They live in high elevational areas around 8,000 feet, but they have been found as low as 6,000 feet, which is still the highest elevational range among mammals. They have adapted to live in very cold environments, both physiologically and behaviorally. Their physiology is quite unique because their basal body temperature is 104 degrees Fahrenheit, which benefits them throughout the winter, but may have potential implications when it comes to climate change and rising temperatures. They're not able to regulate their body temperatures when the outside temperatures are high, but not even that high, like 77.9 degrees Fahrenheit. And if they're exposed to this temperature for more than a couple hours, it can result in heat stroke and even death. So this is where their behavioral regulation comes in. During the heat of the day, pika will retreat to the talus slopes and the rocks to protect them from the elements, because they also hide here during the winter to stay insulated by the snowpack. And climate change will play a really large role in the pika's success, as warming temperatures are reducing the amount of suitable habitat for them, and the reduced snowpack and or earlier snow melt will reduce their range and reduce their ability to survive. So the pika has been petitioned for the Endangered Species Act before in the early 2000s, but it was concluded that climate change was not going to cause any immediate extinction within the foreseeable future, and thus they were not listed. Unfortunately, the Endangered Species Act is more of a reactive program, so only goes into place when species are really, really in need, and they're not a very proactive program, prevents them from getting to that place. So given the rate of climate change acceleration and the trend of decline in the pika's populations, it is very important that we continue to monitor and reevaluate the pika's status. And I promise that is the end of my political input. And as if climate change were not enough for these little guys, there is a potential impact of the plague in some isolated areas. And this is where my project comes into play. There is very little known about how the plague is affecting the pikas. It's kind of an area of new research. There has been some pika carcasses found that did test positive for the plague in Lava Beds National Monument, which is very close to Lassen National Park, and thus why this project is happening. So first off, I just want to say this project is still technically a pilot project, which just means it's early on in its implementation, and the overall design of the study is subject to change as we find out what works and what does not work. So the main driving hypothesis for this study are that the small mammals that live in association with the pika are the ones transmitting the plague to them through entering their sites and leaving fleas behind, basically. So what is the plague? The plague is caused by a bacterium called Yersinia pestis, which lives within the fleas, and then the fleas bite the animal that they are living on and transmits the disease to them. So that's why our project is focused on trapping pika, but also small mammals in association with the pika as well. So some potential culprits for these small mammals that are spreading the plague to the pika include wood rats, Neotoma species pictured here, mice, Paramiscus species, chipmunks, Tamias species, pocket gophers, Geomyidae species, and ground squirrels. I'm not going to pronounce that species. So the main goal of our project, like I said, is to trap pika and small mammals in association with the pika, take measurements such as length and weight, ear tag them, collect fleas, get their age class and their sex, and then to also send those fleas to the lab to be tested for the plague. And also we want to use the plague vaccine for both pika and other small mammals that we catch. We won't go into too much detail about the vaccine, but if anyone is interested in the specifics of how it works, I would be happy to explain in more detail at the end of the presentation, and I will share my contact information with you if you're interested. And then lastly, we want to determine the base levels of plague in the Lassen population and assess the efficacy of the plague vaccine in pika. It has been shown to be effective in other mammals, such as the black-footed ferret and the prairie dog, but the vaccine has only been used in one study in one other place on pika, so this is really groundbreaking stuff here. All right, so how do we actually do this? Well, we had 8 trapping sites total, and last year they were scouted for pika sign. And so that these areas were our target areas because we knew that pika were living there. Each site was 100 by 100 meter plots, with the exception of site 3, the weird looking one, but I'll explain that in a minute. And there were 100 Sherman traps per site with 10-meter spacing. And in addition to these 100 Sherman traps, we placed 20 tomahawks around the site at the targeted picketed areas where we saw a sign of their location. So this means latrines, where they're going to the bathroom, and hay piles where they're storing their food. And what we ended up doing with site 3 is it was kind of tricky. So we tried to make it that elongated shape, but we ended up getting cliffed out on either edge. So we actually moved site 3 down across the trail. And so we were able to make it that nice square. It's just not updated on the map here. And so the Sherman traps that I just mentioned are these small metal boxes in the right corner of the picture here. And they're used for the small mammals we were trapping, such as mice, wood rats, chipmunks, and squirrels. We line these Sherman traps with polyfill, which is just a synthetic insulative material. It helps them keep warm overnight since the traps are left open overnight. And the Sherman traps are baited with sweet feed and peanut butter to attract the little critters. Then we just leave them for a designated amount of time, like I said, overnight or for a few hours during the day. And then for the tomahawk traps, these were specifically targeting the pika. They're good for the pika because they are a little larger and a little more breathable, and they are very temperature sensitive. And we also cover the tomahawk traps in rocks to keep them cooler and place native vegetation, usually lupin, in the traps for food for the pika and also padding. They don't really like that grate on their feet. And so pika are particularly difficult to trap, and it's very important to make sure that the trap is not standing out too much from the environment or they will not go in it. And we did have 2 trapping sessions per day, so one at night, nocturnal, and one during the day, diurnal, because we were targeting different species, such as mice and wood rats only come out at night. Pika, chipmunks, squirrels are out during the day. So basically we would open the Sherman traps, leave them open overnight, let the critters come in, check them in the morning, process those animals, and leave the traps open during the day. Check those, process those animals, and repeat the process all over again every day. So what does processing an animal actually look like? So first we have to transfer the animal from the traps into a gallon Ziploc bag. And this is so we can weigh them more easily with little clip-on weighs that we have. And then we transfer them from the bag into the anesthetizing chamber, which you can see in the first picture on the left here. It's basically just a plastic jug with a cotton ball attached to the lid. And the cotton ball contains a liquid anesthetic called isoflurane, and you squirt a little bit on the cotton ball and it makes them nice and sleepy. Don't worry, we don't use very much, only a small amount, and they're never fully under. They're more just like a little tipsy, if you know what I mean. And once they are sleepy, we ear tag them. As you can see in the bottom right corner, I am ear tagging a dusky-footed wood rat. And after ear tagging, we administer the vaccine or the placebo, depending on the species and the sex of the animal. And we do this as a control. So we are only vaccinating every other animal of the same sex and the same species and the same age class. And really that's just to determine if there's any handling effects that negatively impact the animal and make sure that that's not caused by the vaccine. And next we comb for fleas and collect any fleas that have fallen off in the chamber since they get anesthetized too. And then we take full body, tail, foot, and ear length measurements. And then we return all the animals to their traps and we release them in the exact location where they were found and trapped so that they don't get confused and lost. And then the most important part of my presentation, baby pika pictures. You're very welcome. So after we collect all this information, we put it in an Excel sheet, looks something like this, to organize the data and do some summary statistics. So since this project was a pilot study, its main focus was to collect data, and thus we only have preliminary results, and a repeat of the same trapping protocol and procedure will be required for more robust statistical analysis. So here's a few preliminary results for you guys. We trapped approximately 60 individual animals with a lot of recaptures in the mix there. Trap success was between 5 and 15%, which sounds pretty low, but if you ask any wildlife biologist, they'll be like, wow, that's actually pretty good. And we trapped the potential plague vector culprits that I mentioned. As you can see in the graph, mostly mice, a lot of chipmunks, a couple of ground squirrels, and a few of other species as well, along with one little pika. Some more preliminary results. The sex ratio for all of the species was very close to 50-50, which just means that we caught roughly the same amount of males and females. And the age class ratio, we caught more adults than juveniles, roughly 1-to-10 ratio, which is about expected. And the flea load in wood rat was much higher than the flea load in other animals. So for mice and chipmunks, it was usually 1 to 5 fleas per animal. And on this particular wood rat, we had 32 fleas. 32 fleas. It was actually really gross. But this may have some very important implications to figuring out what species are responsible for transmitting the plague to the pika. But again, this was a very anecdotal evidence. So we will need to do more trapping to know for sure. And then there was fire. I would have more results to share if it had not been for the Dixie Fire. Our whole project and lives were put on hold as soon as we heard the news that the fire had reached the park. Needless to say, my project was halted and all of our focus was shifted to the fire and preparing for evacuation. And within a couple of weeks, we were evacuated and I was sent to Crater Lake National Park, where I am now helping with their wildlife crew. And through all the chaos, one thing was made very clear to me, that climate change is very real and it is happening directly around us. Timber is drier than it has ever been. Fuel loads are higher than they have ever been. Fires are burning larger and hotter than they ever have. And this is why it is so important that we continue to study the effects of climate change and then do something about it. Not only for our own safety, but for the wildlife and other natural resources that we've vowed to protect. Save the pika. Just moving forward with this project, I do still have time left in my fellowship. It's not over yet. Obviously, the current Pika and Plague project is put on hold until next year, but I am learning a lot of new skills and helping with the Pika and Peril surveys here at Crater Lake, which is awesome. And as soon as we can get a hold of my data, which is stored on computers, which were locked away, evacuation, as soon as I get a hold of my data, I can continue to analyze and produce more results. And as well as update the procedures and protocols to prepare an incoming fellow for the next year. And then finally, I will be writing a report, which will include discussing the potential impacts of the fire on the study moving forward. So thank you so much for listening, and a huge, huge thank you to all the firefighters on the front line protecting our park right now. Thank you so much to Jessica Johnston. She has been amazing every step of the way and cared about my success so much. Thank you to all the other ESA staff and the Scientists in Parks National Park staff. And huge thank you to my mentors, Mike and Jason, as well as the Crater Lake folks for taking us in. Special thanks to Sean, who has also acted as a mentor for me. And thank you to my friends, family, and my partner for always being there for me and supporting everything I do. And I will now take any questions you might have, and I will do my best to answer them. Here is also some useful sources if you want more information. And my email is jtooley27@gmail.com. So you can email with me with any questions that you have after the fact. And yeah, thank you so much.